Let me say this over the recording. Tomorrow you're going to have a quiz on this material, so make sure that you're ready for it by paying attention. It's material that you've had reading on for the last, I don't know how long, and you had a whole weekend to get it done. It was due on Monday, but now I'm going over it. So pay attention. Tomorrow you have a quiz on it. So there are 4 objectives that we're going to cover today. That's looking at ATP. We're going to look at chemical energy. We're going to look at chemical energy charts. We're going to look at coupling reactions. We're going to look at enzymes and their products, both denaturing and renaturing proteins and enzymes, and the effect on the products of those enzymatic reactions. So we're going to take a look at those things today. Before we start, let's think about something that all of you should have learned from middle school, and that's the idea of kinetic energy versus potential energy. And kinetic energy is the energy of movement. Energy of movement. So in any case, kinetic energy is 1/2 mv squared, where v is the velocity. So, and the faster something's going, the more kinetic energy it has. The slower it's going, the less kinetic energy it has. I think that's pretty simple from the equation. Could someone please close the door? Yeah, the potential energy, gravitational potential energy, is mass times gravity times height. So the higher something is, the more potential energy it has, right? It's kind of simple. In the case of a stone on a hill, the higher the stone is on the hill, the more potential energy it has. Where is it going to be moving faster? Here at the top of the hill, or here at the bottom of the hill? At the bottom of the hill, it will be moving faster. It has more potential energy here, but arguably, and not arguably, but straightforwardly, more kinetic energy here. More potential energy here, more kinetic energy here. Does that make sense?
Yeah.
Because as the ball rolls downhill, it's doing what to the velocity? It's increasing. So as it rolls downhill, it's increasing velocity. Anybody that's rolled downhill in a cart, anybody that's fallen downhill knows that you go faster as you roll down the hill. I mean, there's anybody who's been bobsledding, anybody's been doing just about anything where you're at the top of the hill, you're not moving as fast as you are as you start to slide down, and finally when you're at the bottom, you're moving the fastest. You've been on a roller coaster, then you know when you're at the top of the hill on a roller coaster, you're almost at zero, and then when you're at the bottom, you're moving as fast as that thing can move. Now, just before you start to go up again, right? Okay. So that's potential energy and kinetic energy in a nutshell. That should hopefully jog your memories. Question?
What did the variables stand for again?
So the variables are m is for mass, v is for velocity, and h is for height.
Okay. And the g?
Gravity. So actually gravitational constant. All right. The force of gravity is what that g stands for, not the gravitational constant. But we'll move on. So this is a pendulum, right? It swings back and forth.
Yeah.
Right? So when that pendulum swings all the way up here to the top,
That's potential energy.
is potential energy is at max or minimum?
Max.
Max, because it's high up. When it falls down here, where it's at arguably zero height, lowest, potential energy is at what? Zero height, h is 0, so what's potential energy equal to? Zero. Now, here, the velocity, when it's up here at the top, the velocity is
Not high.
not high, probably 0, just before it starts to swing back down. So v is 0, so the whole kinetic energy is 0 up here, potential energy is 0 down here. Here, potential energy is the highest, kinetic energy the lowest. Here, potential energy is the lowest, and kinetic energy the highest. So it swings back and forth. They interchange each other. That should be something that you're familiar with from middle school. It should kind of jog your memory a little bit. What we're talking here is chemical energy, not kinetic, not gravitational, but chemical energy. Chemical energy, remember we talked about this when we talked about making bonds, making covalent bonds. Covalent bonds form because the atoms have a lower energy state, because they get a full octet and they're neutral. So atoms want this because they're at the lowest possible energy state. The universe is inherently lazy. It wants to be at the lowest possible energy state all the time. So when a molecule or compound has high energy up here, and it through a chemical reaction ends up as in a product that's lower energy, what happened to that energy that it had when it was another form? What happened to that energy? Was it destroyed? No, it can't be destroyed. It can't be created. It could be transformed. It could just be transferred. It could be a lot of things. But the key is it's lost in the form usually of heat and light, and sometimes it can be transferred to something else. But in any case, the point is it's not destroyed, right? It just gets either transferred, transformed, or just lost to the environment, right? So this is chemical potential energy. How much energy something has it has a potential for energy, to have energy, right? So when a substance moves from high energy to low energy, the energy is released. Whether it's transferred, transformed, or just lost, the energy is released in the form of free energy, and that gives free energy, that difference between its high energy, unstable form to its lower energy, more stable form. All right? That loss that gives free energy. So now we're going to move on. Now here there's that same graph. It's a little more complicated, but it's the same graph. So in most reactions, it's not that simple. You don't just have a little. It's not just a straight line and then a fall off. What you have is an input. You have to input a certain amount of energy to get energy out. So there's this input. We call that input activation energy. Activation energy is the energy that's required for the reaction to start. It's not the energy of the whole reaction, it's just the amount of energy that you need to start the reaction going. Once the reaction starts going, it releases energy. That release of energy is the output. So just like a roller coaster, right? In order for the roller coaster to go, you have to go up the hill. Everybody that has ridden a roller coaster knows you have to go uphill for it to go downhill. Right? Okay, so the same goes with a chemical reaction. If you're starting with AB and you're going to end up with some chemical reaction C, right? Some product C, you have AB and you end up with product C. What do you notice about A, B, and C? Who has more energy? AB has more energy. And if this is energy, which one's higher?
Oh, yeah.
This one, AB, right?
Mm-hmm.
So C is what? Much lower, right?
Yes.
So where'd that energy go?
Down.
It lost. It could be lost. It could be transferred, right? It could be transformed, but it can turn into heat and light. Or, in the case of fire, let's look at fire. If we look at fire, this is what? What does the input represent? Yeah, activation energy is correct. What's the activation energy? When you're going to go start a fire? The spark. Somebody said it's spark. That's right. This is the spark. This is what starts the fire. The fuel is what burns, and oxygen is what is being added to the fuel in order to make it burn. So what's released after the chemical reaction starts?
Gas.
Heat. Gas is released, but heat and light. That's why a fire is bright. There's your output. That's your input and your output, and that's what we're talking about there. Now, there is another possibility. And that's it. Look at this one. What's the difference between reaction type 1 and reaction type 2? That's right. You know what? No one's all day recognized that. I gotta say, that's very observant. A lot of people said a lot of other things which are true, but that wasn't the point. You jumped right to the point. Very good. The product has more than you started with. You have the product here, in this case, let's call it E. E has more energy than CD did. Do you see that? Mm-hmm. E has more energy. CD had, let's say, 10. E has 20. Now you still got some output, didn't you? But really, your output was dwarfed by how much you had to put in. These are called endothermic reactions. And if you want to see one of these, by the way, do you think that this number 2 is going to feel cold or hot? He says hot. Anybody else?
Wait,
Number
what was it,
2.
number two?
Number 2, do you think it's going to feel hot or cold? Why cold?
Because like less energy is released.
Less energy is released, but actually what the key is that look at the energy that's released. Isn't it, aren't you putting more energy in than you're getting out? So what does that mean if you're putting more energy in than you're getting out? It's taking energy. Where's it going to take energy from? Well, if you're touching it, it's going to take it from you, right? So whatever's touching it's going to take. Somebody said transform and what was it? Transfer. So heat's going to be transferred from you to it. And in fact, these are called exothermic reactions. They generally feel hot. They release heat and light. And these are endothermic reactions. They absorb energy. They absorb heat. Thermic meaning hot, heat, and endo meaning in, going into the thing. Exo meaning out, and thermic meaning heat. So in reaction type 1, you're releasing more energy than you put in. That's usually releasing heat and light. And number 2, it's absorbing more energy than you're putting in. That's usually heat that it's absorbing, so it feels cold. Are we clear? Yeah.
In the case of the fire, the endothermic reaction, what would the...
A fire is not an endothermic reaction. How do I know that? Fire's hot. It's releasing more energy than you put in. You put in a little baby spark and you get a big fire. Was that endothermic or exothermic? Exothermic. You got more out than you put in. That's definitely exothermic. Okay?
In the case of the fire, what would be the exact moment when the little top hill, what would be that part? What would that...
As soon as the fire starts, that's this part. So when you're sitting there rubbing, for instance, let's say you're rubbing 2 sticks together to start a fire, you're rubbing, you're adding and adding heat. That's this part right here. That's the input. Then once that fire catches, that's this part where it's releasing all that. It's just a chemical reaction though, that's the point. Okay, let's move on to the next part then. Let's take a moment and look at ATP. Again, you should have looked at this over the weekend. You should have cut them out, the little ATP molecules. You should have put them all together. This should be all review for you. ATP is adenosine triphosphate. Adenosine is a nucleotide. It has the ribose sugar, the nitrogenous base, right? In fact, there's an ATP, there's a GTP, CTP, TTP, and UTP. Where am I getting those?
Internet.
ATP, GTP, CTP, TTP, UTP. Nucleotides, DNA or RNA nucleotides. There are triphosphate forms of all the nucleotides. ATP is an A version of it, and it's the most commonly used in your body. It's for cellular processes, period. So what we do is in fact, to make a DNA or RNA, you generally have these nitrogenous bases charged. We call them charged with phosphate units. 3 of them, hence tri, right? Okay. So, and, a little schematic, so you don't have to draw this. You can draw just a little nitrogenous base and then 3 phosphates on it. Of course, you have the sugar. I should put sugar here, nitrogenous base. So sugar, nitrogenous base, you add 3 phosphates on it. That's what we're dealing with ATP, right? So ATP is very high energy. It takes just a little bit of a bump, and then you get a lot of energy out. That's all that gives free energy you can get out. So that energy can be used by your body. Your body uses this for cellular work. This is what we call currency of the cell. So look, if I was going to hire you to do some work, I'm moving and I want you to help me move, which I've hired students before to do this. So if I'm hiring you to help me move, and I used to live in the neighborhood, now I don't live in the neighborhood, but where I taught. But if I'm going to hire you for work, I actually went to the football team and I asked them to do the work for me because they're the football team. What do you want? So I said, here's $20 each. Help me get this stuff done. And they did it. They did it like really quickly, a few minutes. Why did they help me?
For the money.
The money, currency. Do you work if you're not being paid? No. The cellular processes are the same way. If you don't have ATP, you don't get anything done. Your cells have to move molecules from one side to the other. They got to move vesicles. They got to secrete insulin. They got to increase growth hormones. They have to digest food. They have to break things down, build things up. All of that needs energy. Bless you. All of that needs energy. That means you need currency. You need something to pay for that work. All that work has to be paid for, and this is it. This is your cell's dollar. This is the greenback that your cells work on. All right? Without ATP, there's no work going to get done, and life ends. Next, we'll be talking about coupling the reactions. Coupling reactions is how you get the other chemicals reactions to work with this ATP. Let's take a look at this. This is the problem that you're going to get tomorrow as a quiz. One like it. Not exactly like it, but I hate when that happens. Oh, sorry about that. Listen, one more complicated look at this energy chart. So a chemical reaction, what is it? You got a chemical reaction, you have this beaker or a cell, right? And there's just billions of these molecules floating around, and they have to find each other, right? You got to find the A has to meet B to make C. You got to get A and B to come together to meet C. Okay? How do you get A and B together if there's billions of them together? It's going to be random. They're going to randomly meet. How often they're in a room full of billions of people are two people going to meet? How are we going to get, it's all random, so it's just bumping into each other. So if Meltzer and Curie were destined to be together, and you have a room full of millions of people, what are the odds that just randomly Curie and Meltzer are going to meet each other? Not very good. Not very good. How can you increase the odds of Meltzer and Curie to finally find their soulmate?
Killing the people.
Well, there's two ways we can do it without anything else. There's two ways we can do it. Just off the top of my head, I can think of two ways. We can increase the number of Meltzers or the number of Curies. So, in a minute. So we can multiply, make 500,000 Meltzers and 500,000 Curies, put them in the same room, and it's probably likely that they're going to meet, right?
No.
So we can clone them, in other words. Just clone them and send them in a room, and they'll meet each other, and all kinds of love will happen, right? All right, so the other way we can do it, the other way we can increase the odds of Meltzer and Curie meeting each other is increase the kinetic energy. We can increase their movement. So everybody start moving and keep moving till Meltzer and Curie meet each other. That's it. Increase the amount of movement. And so those are two ways we could increase the temperature. By increasing the temperature, we're increasing the average kinetic energy. And that makes it more likely that Meltzer and Curie will meet each other and life goes on. The other way is increasing the reactants. Now, neither one of these ways really works well for us in cells. If you want to increase the amount of, if your reactant is sugar, it's not a good idea to increase the concentration of sugar in the blood or in the cells, right? Because that's just going to be a bad thing. We call it diabetes if it gets too high, right? So increasing the reactants is often not possible in a living cell because there are other consequences. Increasing the temperature is also not a good idea because when you increase the temperature, other bad things happen, including, so with some of the reactions, they might have to increase the temperature so high that you burn up. And you don't want to end up burning up just so that you can fall in love or solve a math problem or move across the floor, right? You don't want to end up catching on fire. That's just not a good option. So increasing the temperature and increasing the reactant concentration, although to some extent works, is not the best way, is not the way that living systems choose to increase the chemical reaction rate. The way that living systems choose to increase the speed of the reaction is to use a catalyst. And the way a catalyst works, it lowers the activation energy. So where it used to require this much energy to do the work, now it only takes this much energy. So back to our Courier-Meltzer example. I know nobody does, but it's the best one to get people to understand this. So Kemp can go and grab Meltzer by one hand, grab Curie by the other, and bring them together. Now Kemp is not going to get involved in the reaction. Kemp isn't going to marry anybody, but she can bring them together and start the reaction going, right? And then she's off to find another Meltzer and Curie to bring together.
So she's that spark.
She's not the spark. She's the one that's bringing people together. She's bringing people together. That's what she's doing. So at catalyst, and in a biological system, a catalyst is an enzyme. So enzymes bring reactants together. So in this case, AB is brought together with C, and then what happens is A, C, and B get produced. But notice that the enzyme is not involved in the reaction. The enzyme's just bringing the two reactants together in these billions of little atoms or molecules. The enzyme, it's a biological catalyst. It brings these molecules together. An enzyme, and write this down because people all day have not been understanding this. Enzymes are proteins. They're proteins. Now, some of them are RNA. For instance, a ribosome is an enzyme, but it's made of RNA. But mostly enzymes are proteins. Am I clear on this? Okay. So these enzymes, they make products. They make it by bringing them reactants closer together, and they're dependent on shape. You've heard Mr. Desipeta say it. You've heard me say it. Shape and function are intricately related. So the shape of the protein, you can see here's a crazy shape that I made, is going to determine the function. And what you get is this is one reactant, this is another reactant. So this is Meltzer and this is Curie, however you want to look at it. So you have reactant AB and you have reactant C, and they fit perfectly into their slots. And then the enzyme folds and brings them together, and they meet exactly the way they're supposed to meet. Like two puzzle pieces in a jigsaw puzzle. And as soon as they match, the enzyme releases them, and away goes the couple. Are we clear? But understand that this enzyme is not involved in the reaction. The enzyme is not used up. It's not involved in the reaction. The ABs are going to start going away. The Cs are going to start going away because now you're going to have a new combination, right? That's not AB, it's not C, something in between, right? It might be A, B, C together. It might be something else. Whatever the products are. These two are going to be brought together. Something's going to happen, but the enzyme is not involved in that reaction. The enzyme is just putting them together. Is that clear? It's lowering the activation energy, making things happen faster. That's all. Enzymes also are very, very specific. An enzyme works on its reaction and its reaction alone. For example, one of the enzymes we studied all year is DNA polymerase. DNA polymerase only makes DNA. It doesn't make RNA. It doesn't make proteins. It only makes DNA. It's very specific. RNA polymerase makes what? RNA. It doesn't make protein. It doesn't make DNA. All right? A ribosome makes what? A ribosome makes what?
Protein.
A protein. It doesn't make DNA, and it doesn't make RNA. It's very specific. All right. So now, there's a really interesting effect that happens when you heat up a protein too much. If you heat up a protein to the point where it loses its shape, oh man. If you heat up a protein, all right, it could be an enzyme, it could be an RNA, usually a protein, but sometimes an RNA. If this enzyme is heated up to the point where the hydrogen bonds are broken down, it unfolds. it turns into something that looks like spaghetti rather than its shape that it's supposed to have. And therefore does not function. It does not work because it lost its shape.
If you heat up the what too much?
If you heat up the enzyme too much, it loses its structure, it loses its shape, and therefore it loses its function. Very important to answer the question you're going to be asked today and again tomorrow. So if you increase the temperature, we call it denaturing. It's denatured. And by denaturing, I mean it's no longer its natural form. If a protein denatures, then it loses its function. Now, if you cool it down quickly enough, it'll renature, it'll form its hydrogen bonds again exactly the way it was, and it'll keep doing its job. So that's very interesting. One of these things, one medical application of this foundational concept is that you can have a really bad fever as a child or as an adult and be taken into the hospital with 105-degree temperature. And your normal temperature is about 98.5, something like that. But if you're at 105, 106 degrees, and you walk into an emergency room, they might think it's important to get you into ice right away. They could take you and put you in a bathtub full of ice. Why are they doing that? Drop the body temperature so they can renature your proteins, renature those enzymes so that you can keep functioning. Because without those enzymes, you don't function. You're dead. That's what we call dead. So we have to get the temperature down so that your enzymes can renature themselves. All right. So now when we take a look at this concept here, let's look at an experiment that looks at this. So you have this chemical reaction, A plus B yields C. C is a gas. What do gases do in water?
Evaporate.
What do gases do in water? They make bubbles. They make
I
bubbles.
said that before you said it.
Interesting. So gases. So this reaction only happens in the presence of an enzyme. Without the enzyme, this reaction rarely occurs. Won't make very many bubbles at all. Who's ever put hydrogen peroxide on a cut? Okay. Hydrogen peroxide doesn't bubble very well by itself. If you look at it, pour it in a glass, there might be a couple of small bubbles, but for the most part, there's no frothing or bubbling. You take some of that hydrogen peroxide, put on a cut. In your blood, there's something called peroxidase. It's an enzyme that breaks down hydrogen peroxide. And what that enzyme does, it starts to make bubbles. And that's why you see bubbles when you pour it on a cut. All right. So in any case, if we have a reaction vessel here, and we have some kind of container that holds this water, all right,
That's the container.
it's holding this water, and this is an inverted test tube, and it's full. It starts off full of water. This tube here goes from where the reaction's occurring. Notice it's sealed theoretically, right? It's a bad sketch, but we'll go with it. And here's a rubber tube. It goes into the container. Water can go in and out here. All right, so maybe that's badly drawn as well. Mr. Creary, could you pay attention, please? It's not funny. I'd really like to get on with this. So the water's moving and it can move in and out. As the bubbles fill this container up, what is the air pressure going to do to that water? Air is going to expand. The gas is going to expand. That's right. So what's the water going to do? It's going to move out. It's going to move out. What does it bubbling up mean? I don't understand some of the comments that are coming out. You're not thinking. You can't possibly be thinking. So what the water is going to be doing is that the water is going to be pushed down as the bubbles go up and fill up the container. Does that make sense? Yeah. So I can measure the volume of gas, and that could tell me something about the reaction, right? We understand this experiment? All right, this is an experiment that's done often. I've seen it many times on different standardized exams. It's been given to us as a released answer from the last end of course exam. So you have to understand these concepts All right. So they're going to give you some kind of setup. You're going to have to think your way through the setup. I don't know what kind of question they're going to give you. So you have to think, what's going on? What's happening? What controls this? Right? So here's this reaction run by this enzyme making a gas. The more gas it produces, the faster this reaction goes, the more gas you make in a shorter amount of time. Right? Okay. So here's the data from 3 different experiments, 3 different trials. The first trial is at 20 degrees Celsius. You make 30 milliliters of gas. The second trial is 30 degrees Celsius. You make 20 milliliters of gas. The last trial is at 40 degrees Celsius. You make none. It doesn't work at all. What's happening? Yeah.
The enzyme is losing its shape.
Perfect. Someone paid attention. Thank you very much. And that was Ms. Kemp, for the record. So the enzyme is denatured because the heat that loses its shape. That's correct. So it's not working. So it's not producing bubbles. So it doesn't produce the gas. So there's 0 gas produced. That's the answer you got to be. That's the way you got to think these problems through. All right. What made this not happen? What made it not happen is the enzyme. Something must have happened to the enzyme.
Overheated.
It overheated. Now, if we lowered the temperature again, what would happen is, assuming we didn't cook it, if we broke the covalent bonds, the enzyme will not go back to its original shape. But as long as we did not break those covalent bonds, we just denatured it, it'll renature and reform its shape and continue functioning.
Got it.
Our bodies will die.
Say what now?
If it happens, our bodies will die.
If what happens?
If our cells be denatured.
Cells being denatured is a whole other story. I'm talking about enzymes denaturing. When we break down cells, it's much harder to rebuild the cell once it starts to break down. All right. But an enzyme that breaks, without enzymes, you don't work. You not working, we call dead. Okay? When you don't work, it's called death. All right, let's move on. The last thing that we need to talk about, and let's go through the list here. So we have ATP, we've talked about it. Chemical energy, we've talked about it. You should know the charts, you should know what ATP is, you should know how it works. We've talked about enzymes and how they work. We've talked about denaturing and renaturing. We've talked about the products and the fact and how it works. We talked about speeding. How do we speed up a reaction? How do we slow it down? We've spoken about all that. There should be no questions. This is one thing we haven't spoken about yet, and that's coupling reactions. We're going to talk about that now. That's the last thing on the agenda today, and then once that's done, tomorrow we'll have a whole other set of objectives. You're going to have an activity for you to take home over the weekend. There'll be about 2 videos you're going to have to watch over 3 days and make sure you get the work done. Remember, we only got this week we're only going to have 3 days by the time we're done. Some of us, we don't even have that because they're testing. Next week, we're going to have only 4 days. Is it 4? Yeah, 4 days. Because one day is parent-teacher conference. All right, so you'll be here only 4 days next week. And then you're on spring break. Now for spring break, you have another big packet. It's going to be big, and it's going to cover ecology. I'm not going to go into the whys again. I really don't have time for that. Seriously, I don't understand why you ask why. Yeah, I'm just going to get, anyways. So anyways, the point is that we're going to, the why is kind of a ridiculous question because as I said to you before, you have a pro-core exam when you come back from spring break. And you got to know your environmental science, your ecology. And next week, Monday or Tuesday, you're going to have a respiration photosynthesis exam. And then you're going to have to start going over ecology, biomes, different ecological processes, niches, ecological roles, forest succession, different levels. You're going to have to do a project. I mean, there's nothing else we can do if you want to be ready for your pro-core, which is the week, that week you get back from your spring break, you're taking your pro-core. And then you got a month to review everything and to go over everything one more time, and you're taking your end of course exam. Do you want to be ready for these tests or not? Well, then we gotta go over it. That's
Put
it.
in the work.
All right, so objectives, coupling reactions. So when we look at chemical reactions, we take a look, let's say glucose, and we want to break glucose down. Glucose is fairly stable. You can sit it on a shelf, you know, it's probably in your shelf somewhere, sugar, you have somewhere sugar on your shelf somewhere, it's not breaking down, it's just sitting there. Well, we have to take that and break it down. And so what we can do is through various stages of intermediates, which I'm not going to get into, we can take and we can couple it to ATP, the breakdown of ATP. ATP, as we discussed already, is high energy, right? ATP has 3 phosphates. And we can take and it can come in, and out comes ADP. So ATP had A, right? And then it had 3 phosphates, right? And out came ADP, which is again A with what? 2 phosphates. What happened to that 3rd phosphate? It was used. What it was added to this thing. I'm not going to give you a name. I'm just going to say it's an intermediate. And what happened was you added a phosphate to it. So it was fairly stable, but now you have a molecule that has become unstable, has become energetically unstable. So it now wants to release this phosphate, it wants to change, where before it was willing to stay the way it was at the temperature, at body temperature. Now we call this coupling reactions. This is how ATP pays for things to happen. If you want something to happen in a cell, ATP has to come in and give a molecule a phosphate, and once that happens, that molecule becomes high energy and is able to change. Without that phosphate attached to it, it can't change. And that change is what drives the various chain reactions that occur. Ultimately, glucose is changed to pyruvate, and we'll talk about that next week. And you'll go over it over the weekend, because that's going to be your project over the weekend is going to be respiration, finishing photosynthesis and doing respiration. So pyruvate is a 3-carbon molecule that goes then into the mitochondria, into that thing called the mitochondria. And in the mitochondria, you break it down further, and that's where you start producing CO2. And you produce a lot of ATP, and there's all kinds of other things that happen. We're not going to go into much of the detail, but we're going to outline it, and then we're going to move on. Are we clear? Is this all understandable? Is this stuff that you're going to be able you're going to have to work on it tonight. You're going to have to make sure you know it, because there's going to be a major quiz on it, and tomorrow we're going to finish up photosynthesis. So make sure you have all that photosynthesis packet complete. It was due on Monday. Today is Wednesday. You had a whole day with nothing to do but that packet. If you didn't do that, shame on you. You have tomorrow. We're going to go over. We're going to go very quickly through photosynthesis as a review. If you don't have any questions because you haven't done the work, you're going to pay for it in the end. And there's no time for me to go back. So we're done. All right, seeing no questions, we're done with this video. You can start wrapping up and going over your packets now and ask me individual questions.